Hello?
Hello. And I can move over a bit. You can see my beautiful squash. See, there you go.
Great. That's great.
I've been doing a lot of work around the house and not much research.
Oh, yeah. You're probably still recovering from your eye surgery, I imagine.
Well, they got me my glasses back, and these work.
Okay.
So I can start research like I could have started last week, but like I said, I'm playing whack-a-mouse. So.
Oh, yeah. Yeah.
I will try to get started.
Okay. Yeah.
Hello.
Hi, how are you?
Okay, I just finished practice.
Oh, good. So, yeah, welcome. Hello, Morgan. Yeah, so I guess Dick sent me a talk he wants to practice giving in the group here and get feedback. And he sent me the slides, so I have those open. So,
okay, then I'll say next slide, please, when you're ready.
Yeah, that would work. So let me open it up here. Hello, Morgan. I'll share my screen, and then I'll just advance the slides when you tell me.
Okay.
All right. Let's see. Okay. All right. Can you see?
You want to wait the regular 5 minutes to see if anyone shows up?
It's about 9:03, so or 3 after.
Yeah, I know.
Yeah.
Okay.
All right.
Here.
So this is the presentation next Thursday.
Okay. And yeah, this is the paper that you and Rob Stone wrote together, right?
Yeah, we did 2 papers in that issue.
Okay.
Well, he did one. We did a tutorial on Janus-faced logic, and we also did I did a paper by this title, except it didn't have the part after the comma.
Okay.
Okay.
Yeah.
Since this is a memorial symposium, Lev Belusev died a few years ago. I decided to check out his works, and it looks like he did some Janus-faced stuff. Okay.
Okay. Yeah.
Okay. Next slide, please. Okay. So, what I want to do is make a contrast between phylogeny and embryology. This is the tree of life. This is sort of a standard one. There are many alternative views of it, but basically everything is branched, and it's a simple bifurcating branch. Okay.
Okay.
With all the organisms, bacteria, archaea, and eukarya being the 3 domains. Okay. Next, please. Okay, here is a differentiation tree of the early axolotl, Endosperm mexicanum, and it shows how various tissues that we've named over time have been are organized, and our hypotheses about expansion and contraction waves, all of which have been at these stages observed. Okay. So this is up to neural plate formation, basically. Okay, and the way the tree is organized is if let's see, if it's left, it's got an expand. Oh no, I got the see, I found the mistake already. Okay, if it's left, it's a contraction wave, and if it's right, it's an expansion wave. I'll have to fix that.
Yeah.
Okay. Okay. So I colored them red and green for the 2. Okay,
I see the green ones. Well, the green ones go right, correct? Or?
Green is right, and that's the expansion wave.
Okay.
Okay. and left are the contraction waves, so they're red. Okay, so we got all these tissues organized in this fashion. These are based on observed waves on the embryo. The numbers are kind of weird. They're 0 means contraction wave and 1 means expansion wave in these long digits. And the first number is how many steps it's gone through. Okay, so you don't put trailing zeros on numbers like these.
Yeah.
Okay, because it has a specific meaning with a specific number of digits. Okay, so 4 has 4 digits, 5 has 5 digits, so it's gone through 5 stages of differentiation. If you take the middle one, somites, 5 stages of differentiation, and the cells that became somites have gone through, let's say, contraction waves, expansion, contraction, expansion, contraction. Okay?
All right.
So that's what the differentiation code is. Okay, next slide, please. Okay, this is an example of the ectoderm contraction wave. Now, this thing is about 3 micrometers wide. Oh, excuse me. No, it's no 3 microns wide. 3 microns wide. Got it. Yeah, I got the scale wrong. It's 3 microns wide, and it travels in the middle at 2.8 microns per minute. And I think it's faster at the edges. Yeah, 3.7 microns. Okay? And it starts at a point, and it focuses to a point. And it's a dent or a furrow in the surface of the embryo, which travels for some reason on this weird trajectory. It starts circular, becomes elliptical, breaks into an arc, and then closes down. Okay, so that's an example. That's the ectoderm contraction. So you can see it's on a 2 millimeter. The axillary embryo is quite large. It's 2 millimeters. And these things you can actually spot these because the cells contract in the contraction wave, and you can, with some practice, you can actually see that because the cells are pigmented, and when there's a contraction, they're closer together, so the wave looks darker than the rest of the embryo. And we use embryos which are lightly pigmented so that the change in pigmentation is obvious.
Right.
Okay. Okay. Next slide, please. Okay. Now, going back to the phylogenetic tree, it's actually more complicated because there are what are called lateral or horizontal gene transfers between organisms. And this is a schematic view of the tree. Now, what I've done here in the next slide, please. Oh, no, excuse me. Okay, now it's in the next one. Okay, here, I just want to point out that in embryology, it's been dominated by the idea that induction, where one tissue causes another tissue to change type, has been the dominating idea for over a century. Okay, and this is an example of it. Hans Spemann got the Nobel Prize for this work.
Yeah.
Okay, next, please. That would be in the 30s, and he started the work. Oh, I know, early 1900s. Now, if you take that tree of life and we erase all of the vertical branches and just show the horizontal branches, you get a mess. And if that's all you can see, it'll be very hard to analyze what's going on. So it's a phylogenetic tree with horizontal gene transfers, but with the tree removed. Okay.
Yeah.
So it's kind of weird, and if you could only see that, you'd be dumbfounded what's going on. Okay, next, please. So what I want to suggest is that embryology is in kind of that cis state. The molecular development of biology assumes that genes control development. And all the somatic cells in the individual contain the same genes. The sex cells would have half the genes. Okay?
Yeah.
Then it assumes different control genes control the development of different tissues. And the problem is that what controls the control genes is not discussed. And so number 3, as it stands, contradicts number 2, that they have the same genes, and the stories that's illogical. Okay, and this is basically the current thinking for most people of developmental biology, and they don't understand that it's illogical to both think that all cells have the same genes, but different genes are turned on in different tissues. But what turns them on is not, just don't discuss that. Sort of magically, some people say the environment does it, or the environment of the cells, or the contact of cells, or the induction of one cell by another, one cell type by another. That's so it's just not a logical system of thinking. Okay, next please. Okay, now this is from stuff we did. Bradley and I did together. Bradley recognized this. If you take C. elegans, every cell has daughter cells except for some syncytial tissues. Syncytial meaning that the cells fuse together. Okay, so except for those, each cell has a daughter cell. Next, please. And the result of that is that you can do a lineage tree where each cell can be represented. Okay? Mm-hmm. Because each cell gives rise to 2 cells. Okay, next, please. And the nematode. It's actually quite a few because they get about 1,000. What's the exact number?
959 in the hermaphrodite, and I think maybe about 1,000 something in the male, which is because they have
I don't know which one is depicted here. It didn't say.
Probably the hermaphrodite, because that's the most common. So yeah.
Oh, okay. So you can see it's bifurcating. And this is a fairly complicated tree, but each bifurcation represents division of a cell into 2 cells. Okay, so that's why the endpoints there's so many endpoints, which are the final adult cells. It also means that there are many intermediate cell types on the way to producing the adult. Okay, next, please. Okay, now if you look close up at one branch of a regulating embryo, and regulating embryos are embryos which have tissues and within which each cell seems to be the same kind. And that's if you do that, there's of course still a lineage tree, which is represented by the fine lines. Okay, so each cell gives rise to 2 daughter cells. But then we can bundle them according to cell type. So we have the ones on the left bundled together and the ones on the right, and those 2 have diverged. Some cells may be left behind. They're represented by the ones in the middle, and they might be stem cells. Okay. Next slide, please. Okay, so this is an idealized differentiation tree. Real ones may be a little more complicated, but with the differentiation codes attached. And what I've done is put the Roman coin on each branch, which is the Janus-faced object. In other words, what seems to be going on is that at each each time there's a division of cells, it's done in a group of cells, and that group has been given a different color. And that's why the arrows are all different colors. And what's going on is the cells in that whole group become one type, but they're organized by an expansion or a contraction wave. So they're organized from above. So in a way, what the Janus-faced structure means is that we're looking down to the cells. The cells create the waves, which in turn cause the cells to change type. So we have sort of a local global interaction between them. And to give you an analogy, suppose you're driving in traffic and listening to the radio and you change your course because of something that's going on. Then you have a global effect on the individual, and the individual behavior is changed according to the global effect. But the global effect may be caused by traffic flow patterns. And so you get an interaction between global and local, where the local, you as an individual in the car, change your behavior based on the global structure. Okay, next please. Okay, this is the apparatus which we observed in neuroplate, showing the dimensions of the cells. And up at what's called the apical surface, we found a microfilament ring and an intermediate filament bundle around the edges. The cells are connected by junctions, and there's a microtubule mat in between. And what we think is going on is that the cells are bistable. And we call this structure the cell state splitter. That they're bistable, and what happens is that either the microfilament ring contracts or the microtubule mat expands, and as a result the cell becomes tall and narrow, or it becomes flat and squat. And I can see I'm missing from this what goes on with the nucleus. The result of that action sends a signal down to the nucleus, and then the nucleus changes type, changes the cell type. Okay? I'm glad I'm doing this because I can see what's missing.
Yeah.
Okay, next please. Okay, this is just a quote from Lev. He didn't use the term Janus-faced, but it's clearly he's saying the same thing. Okay.
Yeah.
Because it's a symposium honoring him. As I said, he died a few years ago, and I wanted to bring out that he's had similar ideas. Okay.
How do I attend this symposium?
After I'm done, you ask me to send you the URL. So this is Zoom. Okay. Okay. Yeah, I just got the URL today. Okay, next, please. The symposium, by the way, ends around 9:30 or 10 a.m. Our time. Okay. Yeah. So I'm not going to be up all night for it, and most of it's going to be in Russian.
Okay.
Okay. There'll be a couple of talks at the end in English. Okay, what I want to show here is that Lev may have been the first to probe this local global interaction and its effect on differentiation. This is an experiment he did where he took an embryo, cut out a sector of it, and then filled the sector with a corresponding piece from another embryo. That's what that arrow means. And the result of doing this was that the whole surface of the embryo became a whole bunch of neuroplates. This shows two of them. Okay? And so what I'm suggesting here is he screwed up the waves, and the waves did their job, and they made neuroplates. Okay, all over the embryo. So this is a very brainy axolotl, or Xenopus in this case. Okay, many brains.
Yeah.
Okay, but it's an example of an experiment where you intervene with the mechanics of these waves, and the result is an abnormal embryo. Well, he probably thought of these in terms of these bidirectionality things or Janus-faced behavior. Okay.
Right.
Okay. Next slide, please. Okay. You can read this?
Yeah.
Okay. So there are a bunch of questions that remain about the whole thing. What triggers the differentiation? We don't know. It might be. I can say I put something in there. It might be, for example, that in the place where the wave starts, two tissues have adhered, changing the mechanics compared to the rest of the embryo. And I suppose that's typically been interpreted as an induction, but it may just be a simple mechanical effect. And that may lead to an imbalance that triggers the differentiation waves. But that's all speculation right now.
Well, okay, I always thought it was because the blastopore had gotten large enough to stretch the cells.
Well, that may be part of it, but then where it touches on the inside of the embryo, it may fuse to some of the cells, leading to a greater thickness of the outer layer cells, and that may be what triggers the wave.
Okay.
Okay. Then, as I said, the ectoderm contraction wave has a very peculiar trajectory, and we'd like to know if mechanical tensions determine the trajectory of the wave. Okay, so that's a simple sounding question, but maybe Susan will come up with an answer on these days. Does the speed of the wave depend on the local tension of the surface, or at least depend on the strain? We can measure the strain. It's more difficult to measure the tension. Okay, and then the differentiation waves stop. We don't know what stops it. It could be that it's in another tissue that it can't pass through for some reason, but we don't know. Okay, and then what is actually the pathway? I mean, in this case, the molecular pathway from the cell state splitter to the nucleus of the cell, which I didn't illustrate. And this is how the cell state splitter changes the presumably the epigenetics of the cell and makes the cell change into a different type. And then a little more sophisticated, each cell has got a history. And we don't know what on earth that history is stored in from its sequence of differentiations. So that's why I call it remember in quotes. Each cell remembers where it's been. At least that's a guess. In order to get many cell types, you have to remember what you've done before. We don't know what that memory consists of. It may again be epigenetic, but that's just speculation. Okay, transdifferentiation is when a cell type experimentally changes to an entirely different cell type, or maybe wound healing. Transdifferentiate, when we don't know if transdifferentiation supports this idea or contradicts it. That's why I said transdifferentiation could be done by backing down the differentiation tree and coming up a different branch for the cell. Or it could be something entirely different. We don't know. Okay, another thing is the number of cell types is increasing. And we saw in the C. elegans it goes up to 1,000. So is there a transition from differentiating waves to inductions? I'm not saying inductions can't ever happen, but it could be that there's some sort of transition as the number of cell types increase. Again, that's unexplored. Okay, next question. Does regeneration use stem cells, or do they use the differentiation tree? We don't know. Okay. Again, you'd have to follow cells during wound healing or something like that, and see if they're following the differentiation tree or doing something entirely different. We don't know. DNA tends to be linear, and a tree is not a linear structure. So how a differentiation tree is mapped onto the DNA is a good question. And that will also lead to questions like, okay, what kind of gene duplications can this lead to? And how would it alter the differentiation tree if you had, say, gene duplications or mutations or whatever? Okay, now, continuing differentiation is a concept I came up with because if you look at things like bacteria, let's say these cyanobacteria, they're capable of producing 2 or 3 cell types, and that's it. Okay, whereas eukaryotes can produce literally thousands. I mean, the nematodes got 1,000. For mice, the number of cell types has been estimated as between 250 and about 5,000. Okay, again, we don't, not a very strong figure there. But what continuing differentiation is the invention of the ability to produce more cell types in the course of evolution. And so the question here is, can we explain evolution in any sense by the differentiation tree changing over time through mutations of the DNA? Okay. Then we have a fun one. This is due to Donald Williamson, and that metamorphosis, which is, well, for instance, if a caterpillar changes into a butterfly, they're quite different animals. And the hypothesis is that, at least in some of those cases, the transition may be due to the fusion of 2 different organisms rather than one leading to the other. But in this fusion, their differentiation trees are fused. Okay, so we need. Whoops, let me close the door. Noisy dog, just a second.
All right.
Okay, now Donald Williamson wrote a couple of books on this subject, but he did them before we had any sequenced genomes of whole organisms. So I think that there's a possibility by examining now we have thousands of those, by examining some of those where metamorphosis occurs, we could possibly see if the differentiation trees of the organism that undergoes metamorphosis is actually a fusion of 2 other differentiation trees. But this would require some pretty high order examination of genomes.
Right.
And then finally, I'm doing a paper with Bashir Ahmad on this question, and that is, it is generally assumed that by biologists that physics is reductionist, and the Janus-faced causality of embryos that we may have uncovered is not reductionist. But if you start looking at modern physics, you find that many aspects of physics are no longer reductionist. So there might actually be some intellectual similarity, and for a possibility of a new philosophy of science that might cover both embryology and physics. Okay, next slide, please. Okay, now, I was asked to make some personal comments about Lev. So unfortunately, I don't know him well. I only met him twice. But in terms of stories from him, I won't say who, but he was very badly treated by a California embryologist. And then I had some funny situations with him. When I went to Moscow for a meeting he organized, he got me through customs. A friend of mine, I gave her a document that was in Russian, and she said it had to do with visas, and I ended up flying to Moscow with no visa.
Oh.
So I was stuck in the airport for a couple of hours, but I think it was on a weekend even. He got me out. Okay, and then later it's a long trip to Russia, and the time lag is enormous. That's why I don't recommend trying to take in the whole conference. Unless your Russian is fluent, then you can stay awake. Okay. Okay, so since this is addressed to people who were awake, that's why I missed most of your talks.
Yeah.
Okay, and then the funniest thing was the end one. There was embryonics is an attempt to make computers based on embryology concepts. Okay, and there was a conference being held in Switzerland, and we were asked, Lev and I were asked to organize a session of it. We did it. We had half a dozen speakers and whatnot, and then they rejected all the papers, including ours. Okay. Okay, so next slide, please. I'm not naming names and who did these things.
Oh, yeah.
Okay, so this is just a quick summary. Lev and I did a couple of things together, and then our last paper was rejected, but I still have it, strangely enough.
Yeah.
And they might publish it as in the proceedings of this conference.
Okay.
Okay. Next slide, please. Okay, I put together his list of publications and just a little comment there. Okay.
Yeah.
I see I have to change the font. Okay. Okay, doke. Did I miss anybody from the earliest mechanics?
I don't know.
I mean, Benzev, Donald Ingber, George Ostrom. I didn't agree with all these people, but
Right.
but the number of people who were working on mechanics in the early 70s was pretty small.
Right.
And my observations attending the soft matter, I don't know if you're doing that, Bradley, the soft matter conferences.
Yeah.
Is now it's dominated by physicists who don't understand embryology, so they're doing trivial problems.
Right.
Okay. Next, please. They've gotten all the way to the rounding up of a sphere of cells of one type. They haven't even done the onion problem where one cell type surrounds another.
Oh, right.
Okay. This is out of order.
Oh, these are the rest of the questions, right?
Yeah, more questions. Okay. Let's see. I see I've got this slide in the wrong place. Okay. How's it different? Oh, maybe it's the same. Maybe it looks like it's a duplicate, actually.
Oh,
Okay.
maybe. Yeah.
Yeah, next slide, please. Okay, the next is just references. Yeah, that's it. Okay.
Yeah, that's great. Yeah, and then more references and
more references, more.
And then this is the paper here. This is the one with Rob Stone, the 21 here.
Yeah. Oh, wait a second. Would you go through that again? Did I put in the ones with you?
I think in the first slide, right here. This one here,
okay.
yeah.
Is that a decent one? Those two.
Oh yeah, this is the one where we did the C. elegans and the C squirt differentiation trees. So these are differentiation trees of this sort of deterministic evolution or mosaic evolution that we talk about with C. elegans. You see it in C squirts where they don't like it isn't the same as like the type of differentiation tree that you showed there. It's more like we had developed a procedure to look at the cells as they divide and then look at the smaller and larger cells and sort
We
the
did
tree
a
in that way.
differentiation tree, as I recall, of an Ascidian.
Yes. Yes.
Could you send that to me? I was having trouble finding it.
Yeah. Yeah. I have these trees. I have like the entire tree. I can't remember what the link is, but I can get it.
Okay, and the other question is the Google file essentially the same as the paper mentioned there?
It may be. It may be a different. I don't know if it's an earlier version or what. Let's see what this looks like. Oh, maybe it got moved. But yeah, I mean,
Okay.
I can send the paper. It there's like supplemental material, but I think I probably also have that tree somewhere else too.
Yeah, I might want to include the Ascidian tree.
Okay, yeah.
Okay.
All right, yeah, I'll do that.
Okay, I think that's it.
Yeah, so this is great. I don't know if people have any questions or comments.
Okay,
Now's the time.
or threats or whatever.
I mean, I have a couple questions, but I can wait.
Sure, go ahead. Go ahead.
Susan or Morgan, do you want to say anything before I ask my questions?
I already did, just as it turned up. Like, I'm still vague about some of it, but
No, I need to jump into these references and learn a lot more about the area. But
okay,
super interesting.
That paper that got rejected with that Lev was part of, the basic problem the referees had was they had a whole bunch of experiments they wanted us to do, but we had no money. It was extremely hard to get money to do any of this stuff at that stage. Maybe today, now that physicists are getting into it, one could get money more easily. But we put out our ideas, and they said too forcefully. But tough luck.
You list the experiments that they wanted you to do? That would be
great.
Yes. So I'm thinking if we publish the paper, since I have the paper, I still have the referee comments. I'm going to publish with the referee comments as again future work.
Right.
Mm-hmm. Yes.
Okay. Because well, Susan, you know how little money we had.
Yeah, I do.
So
they kept going, well, you can't do that. That's not the way this works.
Yeah.
Earth to people, it probably does work this way.
At any rate, Lev's comments in that paper against positional information are really what I call acerbic. So he made mincemeat of the positional information idea. So, at any rate, and I getting those comments by him now that he's long dead, getting them published is not going to hurt him.
Yeah, that's a good point. Yes.
So, at any rate, that's where we stand.
Yeah.
Okay, Bradley, what are your questions?
Oh yeah, so let me go back to the slides. If I go through, I like to usually go through from the beginning.
You're controlling them.
Yeah. So yeah, this is a figure from the paper that we did, the one from the biology journal. You'll see it in the references there. I can also send it out if people want to read through it. this is C. elegans, so this is where you have cell division, and you have these cells that are deterministic, so you can trace them. You can do lineage tracing, things like that we've talked about in the meetings, where you can actually know, they can put a marker in one cell and it passes down to a certain place in the embryo. And then these eventually are going to form tissues. So AB forms a host of tissues. Things like C and E form different subtissues. And so there's that aspect of it. There's lineage, but there's also the size of the cells. And so you can build something similar to like a tissue-based differentiation tree from the C. elegans. So we did that, and we also did this with ascidians or sea squirts. And sea squirts are interesting because they have like multiple stages of life history. Yeah.
Bradley, I guess another way of putting it is that the lineage tree of C. elegans is also its differentiation tree.
Right. It's very similar. It wasn't exactly the same. What happens
right. Not exactly, because sometimes you get a pair of identical cells.
Right. And what happens is the order of the nodes are swapped. So like what you'll have is the lineage tree as proposed originally was where it was organized anterior to posterior end of the worm. So the cells that ended up at the head were down at the left side, and the cells that were at the tail were at the right side.
Oh, right.
Yeah. So you can see it starts here where you have the posterior end here and the anterior end here. And then there's division and migration, and then the tissues form. So these things get moved around a bit, but that's the way the lineage tree is laid out. Now with the differentiation tree, those nodes get swapped, so the order gets shifted around.
I see what you mean.
Yeah. So that I remember doing that, and like the problem we had there was that we didn't have a very good estimate of cell volume. We worked from some cell volumes that were, collected through some experiments, but those weren't like very good. I mean, you could get better measurements of it. So, but then that's how we did that. And so in a C squirt though, it's interesting. The life history is where you have different stages of the phenotype. So the early phenotype of a C squirt is not this twofold symmetry, but fourfold symmetry, where you have four sort of founder sublineages. Like you have A, B, P1. That's not the nomenclature, but you have two additional founder lineages, and basically it's like that fourfold symmetry instead of twofold symmetry. And then it becomes like later on, it develops a brain, and then it becomes sessile even later on. So there's a lot of interesting stuff, but the early embryogenesis is this sort of, you can start with this simple symmetry, and then it's, the cells are dividing, and you have size information, so you can sort it by size, and then eventually you start to get this tissue differentiation. And so that's where we were doing in that paper. We're looking at that very early stage.
Bradley, I got a question about it. In C. elegans, is the total volume before it starts eating constant or slightly decreasing?
I think the volume I think we determined that it was constant, or at least that's how we modeled it. I don't think there are huge differences in the egg at that stage. So that, the egg eventually expands out a bit, but I don't remember what the scaling factor was. But in the early embryo, it's pretty consistent, okay. And the cells just are like half the size of the.
So they don't have yolk cells?
No.
No. Okay. Okay.
Yeah. So I don't think they do. Well, they have some cells, like some cells that kind of die off. They're not included in the nomenclature. I don't think they do. So, yeah. So that's the one question about the Janus thing. So this is a Janus-faced thing is basically it's a coin, like you flip a coin or it has two faces. So you can treat it as like a coin that you flip or have two sides of.
No. Janus was a god of the Romans that had two a head with two faces, one forward and one back.
Okay.
And was actually put on a coin.
Okay.
Okay.
But
Okay, so one is looking forward to the whole tissue, and the other is looking back to the cells.
so is this a simultaneous thing then? I mean, like, do they kind of?
No,
Oh.
no. Something triggers a wave.
Okay.
The wave propagates through the cells, and as it propagates, it changes the cells to a new type.
Okay.
And then it stops somehow. And then we have a new cell type. Yeah, that's the basic idea.
Okay.
Okay.
Yeah. So then this is the differentiation tree. This would be where you have something triggering a wave.
Yeah, this is an
Yeah.
empirical tree. It's not as neat as the idealized one.
Yeah. Yeah.
Okay. And we try to arrange it here by stages of the embryo on the right-hand side, so you've got embryology time.
Mhm.
Okay, at a given temperature.
Yeah.
Okay.
Oh, temperature is
also-
on the left.
No,
Oh,
I've
no.
forgotten
What
those
is that?
numbers mean.
Yeah, I didn't think it was 50 C, but yeah, that'd be pretty bad. Yeah.
No, I've actually forgotten what those numbers mean.
Oh.
Okay, I'll have to check on that. Yeah, you caught something else.
Yeah.
Okay,
Okay. Yeah. So that's
I
that.
don't know what they are.
Oh. Yeah, I don't know either.
So the red is the contraction wave.
Yes.
Yeah.
Yes, that's correct. The text on the left is wrong.
Oh, okay. All right.
Okay, I have to fix that.
All right.
Okay. Well, I'm glad. Thanks for the opportunity. You found all sorts of errors in this, so I'll try to fix it up for Thursday. And I appreciate if you can send that city and tree today so I can include it.
Yeah, I'll go get it after the meeting.
Okay.
It'd be, yeah, I think it's like in a large format. It's like I think an image format, but I'm not sure if I have maybe some other information as well on
Okay.
that, so I can send it along.
Okay, terrific.
Yeah.
Okay, so any more questions?
Yeah. Well, thank you for that presentation. I mean, yeah. We haven't talked about that in a while.
I would like to go over it in more detail, actually, but
Okay, I'll send you the next version too then.
oh, all right.
Okay.
Thank-
Yeah, if you get up about 8:39, I think I can send you these sessions. There's some sessions which are, I think they're going to be 3 talks in English at the end, which is in the morning for us.
that would work. My Russian is worse than my French, so there we go.
You wouldn't believe it. When I was a graduate student, I actually took a Russian exam and passed it.
Oh wow!
And I had only studied Russian for 3 weeks.
Wow!
And I forgot everything. Okay, I can occasionally recognize the transliteration of a Russian word.
Yeah. So I'm gonna go to some papers now. I think
Okay.
we have something to follow up on from last week.
Okay,
so-
I'll reduce my bandwidth again.
Oh, okay. Yeah, that might be helpful. So Susan actually mentioned something last week about mitochondria, and I mentioned that in textbooks you have this. It looks like mitochondria look like a bean, so they're bean shaped, and you see these things that are sitting in the cytoplasm, and then they produce energy for the cell. But if you actually look at a 3-dimensional scan of the cell, you can see that mitochondria actually form these reticulative networks. So they're connected together. They're these they kind of look like beans still, but they're connected through these strings of things, and they're connected. And so I heard this at a conference, and I was interested to know more. And actually, there's been some interesting work on this. So the first paper I'm going to talk about here is this paper. Let me see if I can get it here. What is the function of the mitochondrial network? A theoretical assessment of hypotheses and proposal for future research. So they kind of review this area of what the mitochondria, how it's sort of structured in the cell and what it's doing. So they state that mitochondria can change their shape from discrete isolated organelles to a large continuous reticulum. So a reticulum is like there's an endoplasmic reticulum in the cell, but it also has this reticulum of mitochondria. And it can actually change its shape. So it can actually have these isolated organelles. It can, at some points in time, it can be like that, or it can have this large continuous reticulum structure. So I assume this means that it's kind of, it can oscillate between these states depending on the function, depending on what's going on in the cell. But that, this is how they envision it, at least according to this view. The cellular advantages underlying these fused networks are still incompletely understood. In this paper, we describe and compare hypotheses regarding the functional mitochondrial networks. These mathematical and physical tools, both to investigate existing hypotheses and to generate new ones, and to suggest modeling strategies. And then the novel insights that they get from the work that's ongoing is that selective mitophagy is not required for quality control because selective fusion is sufficient. I don't know exactly what that means. And then 2, increased connectivity may have nonlinear effects on the diffusion rate of proteins. So this is connecting the different organelles together. And then 3, fused networks can act to dampen biochemical fluctuations. So you might want a network to dampen biochemical fluctuations during certain processes. And of course, the mitochondria produce energy. So there are a lot of fluctuations biochemical wise related to that. So, each cell has a number of mitochondria, and then they get linked together in these networks and they have certain properties. That's basically the idea. So,
Bradley,
yeah,
the word mitophagy, I'm guessing, means that somehow the cell eats bad mitochondria.
Oh, okay, yeah. And so it's not required for quality control because so I guess what they're saying is that only the best mitochondria get fused into this network. So it doesn't require that they get recycled or consumed by the cell. Yeah.
Can I give you a contradiction of this?
Okay.
If you look at some diatoms have chambers in the silica cell wall, each of which contains a mitochondria. And I'm not aware of anybody looking for networks of those. So they might be isolated in individual chambers in the cell wall.
Okay. Yeah.
Okay.
Yeah. So I guess they talk about, like, in cases where you might need to have this sort of structure where they might observe it. Mitochondrial dynamics and structures have been the object of intense study in the past 2 decades. They're fundamental to the function of the cell, highly responsive to cellular state. So in things like Parkinson's, diabetes, cancer, and Alzheimer's, you can have these networks form and change their shape and their connectivity. In mitochondrial diseases, you see this as well. So there are a lot of reasons why these networks form and how they operate. Various effects of fused mitochondrial states have been observed, including an increase in energy production, protection against apoptotic stresses. So these are stresses that sort of force the cell to go through programmed death, or sometimes through necrosis, I guess, as well. An increase in cell proliferation. So if the rate of proliferation increases, you need more energy. and the regulation of various signaling pathways. So they don't know how this, how this is established, but they can observe. So this is where you get the different types of fusion. You get some degradation of dysfunctional parts, sharing of matrix and membrane components. So this is where mitochondria get fused together. So you see this bean-like structure here as a single mitochondrion, and then they get fused together like this. So these are organelles in the cell membrane, and they get fused together. So they start to share matrix and membrane proteins. And then they can form these kind of chains or networks where they're hyperfused states, where they're all fused together in a sort of a shape. So now they're sharing all these materials, and you have this network. And then, yeah.
Bradley, got a question about this. Did they do a phylogeny of this? In other words, when did fusion appear of mitochondria appear in evolution?
Right. I don't. Yeah. Well, we'll see, but I don't think so.
Because it's kind of uncanny, because I'm not aware of fusion in prokaryotes, except for the hypothesized one that created eukaryotes.
Right.
Okay, and if this is correct, it shows that fusion of prokaryotes, at least the ones inside cells, can occur, and therefore could completely change the story of eukaryogenesis.
Yeah, good.
Okay.
Yeah. So and then on the right you have these different types of, they have different regimes for fusion. So this is where you have microfusion, where you have very few of them fusing together. And this is just a change in the parameter value. So microfusion is where you have a few things fusing together. Mesofusion is where you start to get more fusion into these networks. Dynamic hyperfusion is where you get intense fusion, and most of the mitochondria are fused together. And then static, or they kind of move around. So I guess you have these subnetworks that attach and detach in different places. And then static hyperfusion is where all of the mitochondria are in this network, but they don't move around. They're sort of static there. So this is just a matter of, the different types of fusion that they're proposing. And again, none of this is really well understood. It's just kind of what this field is grappling with. And then they have some modeling. So they have some criterion for usefulness of. So I guess these equations here are how useful a mitochondrion is to the cell. If there's a shared usefulness of the I don't know what that means, but basically if it's maybe more energetically necessary to fuse, then they fuse together. And so you can have a pairwise model here where you have...
Bradley, that's an attempt to mathematically say that the whole is greater than the sum of the parts.
Yeah. You're like, how did they fuse together? Well, we'll use a usefulness criterion and then show the... So yeah, that's and then they kind of go through some of the reasons why you have, why you might have fusion. And so, yeah, you have these, it might enable faster energy transmission. It might increase ATP synthesis. So there are a lot of energetic, and Susan was asking about some of the energetic, or she was talking about energetic aspects of mitochondria last week. And I'm going to quickly show that paper in a minute, but this goes through the hypotheses that people have for why you have this mitochondrial fusion, and it's just a list of things that maybe are why you have this. There's some criticisms as well. So these are things that people have proposed these hypotheses, and then of course some of them are more plausible than others, and of course you have all those you have a lot of caveats to these. So it's not we don't know exactly why this happens. It could be multi-causal. It could be, depending on when it's happening or in what cells it's happening in. So that's, and then, yeah.
I can't read that fast.
Oh.
Do any of these hypotheses include the possibility of a communication network within the cell?
I'm looking at them right now. Yeah, they don't really stress communication so much. They talk about autophagy. They talk about maybe efficiency and dysfunction. Yeah, they don't really talk about communication so much.
I know you're interested in non-neurological communication systems.
Yeah.
This might be one.
Yeah, it looks like, because they talk about like robustness too, and it's like robustness in terms of what? Is it like just to make sure that there's enough energy being produced, or is it that they actually communicate or they send information to coordinate some of this energy production? It's so yeah, we don't really know. And then there's actually some hypotheses, but now you put it in terms of further modeling and then experimental tests. So you have, for example, increased selection bias and quality control. And this goes back to where we were talking about where different mitochondria are connected into this network based on their quality or their like level of ability to produce energy. And then you can use ordinary differential equation models to do this. And then that might be a way to model it. If there are other ways to model it, but this allows us to look at like stochastic influences and other things. And then the experimental test would be to look at the assumptions of this example where small mitochondrial fragments are degraded. Only small mitochondrial fragments are degraded. The existence of a threshold size above which a mitochondrial filament is not degraded by macrophagy can be measured. So you can actually look at this quality of a mitochondria experimentally and then look at how they're forming networks and actually see if that's why that's happening. And again, that goes back maybe to the communication issue where, maybe there's some other thing going on. Maybe that's, in that criterion of why a mitochondria might connect to this network. So that's and then so I don't think they talk about evolution in here. They just kind of go through some of these hypotheses. They do actually some interesting modeling here. So this is a diffusion model of the formation of these networks using some sort of I guess a two-dimensional fluctuating lattice. So they're actually it's a lattice simulation where they're looking at sort of like how these things connect together. So, you can do all sorts of modeling to look at this. So this is more of the sort of how we might do the experimental or how these things are fused together, where you have, fusion points and you can have a perturbation to break them apart or to prevent them from fusing together. So they show a lot of the biochemistry involved in that and maybe some of the ways you can experimentally keep that from happening. So I'm going to, in the interest of time, I'm going to go to this other paper. And this is mitochondrial network complexity emerges from fission-fusion dynamics. So this is one example. That was a large, broad review of like what's going on in that area. This is more of a specific question that they're asking. And they're asking this network complexity. And so a number of these people have worked on brain networks or like networks from neuroimaging and things like that. But they're also asking this question of the mitochondrial network. So it's very much a network science question. They're kind of bringing tools in from other types of networks to ask this. And they're asking, how does this network complexity or how does this connectivity emerge? And so they propose it's fission-fusion dynamics. And you saw a little bit of that in those simulations and in those diagrams where they propose different mechanisms for that. In this case, the abstract reads, mitochondrial networks exhibit a variety of complex behaviors, including coordinated cell-wide oscillations of energy states, as well as a phase transition or depolarization in response to oxidative stress. So there are all these dynamics going on within mitochondria and mitochondrial networks. So there's very much that issue of like processing going on, and the question is what is the thing that's sort of shaping these networks? Since functional and structural properties are often intertwined maybe. Here we characterize the structure of mitochondrial networks in mouse embryonic fibroblasts. So this is in mouse, using network tools and percolation theory. So this is percolation theory is a tool from physics where they sort of grow networks, and they look at, percolation is this process where liquid goes through a layer of like maybe like rocks or mud, and it kind of goes through, and you look at how it moves through that medium. So, they use that as a metaphor for sort of looking at how networks emerge, different nodes emerge with connections between them. So it's a different, it's a way to do this sort of simulating the growth of networks. So they actually look at this. They have do some quantitative analysis of mitochondrial clusters. These reveal that structural parameters of healthy mitochondria lay in between the extremes of highly fragmented and completely fusion networks. So you saw in the example from the other paper they had where, you had an example where there were no mitochondria fused. They were all these little beans that were isolated. And then the second example was where there was another example where they were all fused together, sometimes dynamically, sometimes statically. And so they're arguing that what you actually observe is sort of an in-between state where a lot of times some mitochondria are independent and some form these networks. So we confirmed our results by contrasting our empirical findings with the predictions of a recently described computational model of mitochondrial network emergence based on fission-fusion kinetics. And this is just where you're looking at things that are fusing together or breaking apart, and the kinetics behind that. Although these results offer not only an objective methodology to parameterize the complexity of this organelle, but also support the idea that mitochondrial networks behave as critical systems that undergo structural phase transitions. So phase transition, of course, is where you get a critical value where you go from like something that's disconnected to something that's connected. And so these are mitochondrial networks. These are images that they're using from confocal microscopy. They're looking at these cells. This is a YFP marker, and they're looking at how they sort of form. So this is just an example of that. Then they show some examples of things where you're looking at like power law behavior and things like that. So you can identify phase transition from these power law signatures. The exponent of the power law is usually the thing that tells you whether there's like a phase transition going on. And again, it's like where you have this, this state where they're independent, and then there's a rapid transition towards this network structure. And I don't know if there's anything more here, just kind of showing this, how you get these different power law signatures for different types of network topology. So you get these clusters, and then these are the power law signatures for these clusters. You get 1 segments and 1-3 segments that they call. So these are different parts of this structure, and you see different power law behaviors for that. So there are changes in mass distributions upon fission-fusion balance perturbation. So when they look at the different parts of these networks that are fused together, they can see changes in the mass distribution, which is just a characterization of how they're put together, I guess. So this tells you some things about like that emergent property, or how the whole is greater than the sum of the parts. Just ways to measure that. And then they go through some other things. Here's some comparisons of some experimental results with a model. So this shows, just examples of this where you have, this is a phase transition here on B. C is panel C through E, which are down here, show a graphical representation of the typical networks simulated using 3 derived values corresponding to different types of networks, PQT, CLT, and MFN. And I think those are things that they've observed here where they look at the phase transition. So they're looking at different points for the phase transition for the C parameter, and there are different points. And then this shows why those points differ, because the topology differs of the network. So you can see C is largely disconnected, D is semi-connected in different parts, and then E is more fully connected. And you can see that the phase transition is later for each of these. So they move out as they get more connected. So that's what they're doing there. And then finally, this is the paper that Susan sent me. She sent me a paper on the original paper on this. So let's see if I can get this to. Okay. This was the paper here, the energetics of genome complexity. So this is a kind of, she was talking about last week about energy balances and things like that. And this is a paper on looking at sort of molecular energetics. Let's see. This is Nick Lane and William Martin. This is out of, I think, Nature. It's a hypothesis paper. So the abstract is, all complex life is composed of eukaryotic or nucleated cells. So these are eukaryotic cells. They have organelles inside, and they have, of course, mitochondria, and they have a mitochondrial genome and a nuclear genome. And they're very distinct. Mitochondrial genome are specialized for energy production, and of course our nuclear genome is more general. The eukaryotic cell arose from prokaryotes just once in 4 billion years. So this is an event where a lot of different organelles came together to form a eukaryotic cell. The prokaryotic cell doesn't have a lot of the same complexity as eukaryotic cells. This was a sort of a symbiotic event that occurred. Prokaryotes went on their own way after that fusion event that formed eukaryotic cells. So it isn't like a transition from prokaryotes to eukaryotes. It's that eukaryotes, they had this evolutionary event where you had this symbiosis, and then prokaryotic cells never really showed any greater complexity that characterized what happened in eukaryotic cells. And then they ask the question, why not? And then they go to what they do is they look at the genome of the prokaryotes, they look at the genome size, and they say that that's constrained by bioenergetics. The endosymbiosis that gave rise to mitochondria restructured the distribution of DNA in relation to bioenergetic membranes, permitting a remarkable 200,000-fold, which means it's 200,000 times greater than, expansion in the number of genes expressed. So what happened in eukaryotes is with all these organelles that came into association with the rearrangement of the genome and with this nuclear genome that's very diverse, and then a mitochondrial genome on top of that, you had this large number of genes that were expressed beyond the prokaryotic genome, and the genomes were bigger and more flexible. This vast leap in genomic capacity was strictly dependent on mitochondrial power and a prerequisite to eukaryotic complexity. So you needed to have that power source basically in the cell to drive a lot of this complexity. So if you think about mitochondria as being the powerhouse of the cell, having a powerhouse within the cell is actually quite useful, as opposed to like individual cells being in it for themselves and getting their own energy from the environment. And so that's kind of like almost like how embryos form from, just multicellular associations, so that there's that sort of evolutionary transition that I don't know how many, I don't know what kind of work is, other than this is a hypothesis paper, so I don't know what other kind of work has been done on this. But so I think that frames this idea of mitochondrial networks nicely, in that you need these networks for energy production, and what's the most efficient route to energy production.
Okay, Bradley,
Yeah.
a question. In any of the three papers, did they discuss whether or not any other example of prokaryotic fusion has been reported?
I didn't see anything. I haven't read this whole paper, the genome complexity one, but I don't know if they talk about that. It doesn't sound like they have any examples of it because they kind of use it as a contrast.
Because something interesting is going on here. There's no obvious reason why other prokaryotes couldn't do the same thing.
Yeah.
And you have plenty of occasions for doing that, such as in gnats. Okay.
Microbial mats, yeah.
And the gnats go back at least to the beginning. They're archaeal gnats reported by paleontologists.
Yeah.
A few billion years old. Now, in terms of, you might want to ask George Mikulowski to give us a talk.
Oh yeah.
Because he and I did 2 papers on why it took so long for fusion to occur, and we concluded that it was due to having enough variety of genes that were transferred by horizontal gene transfer. I'm not sure we're right, but but he did some calculations, and I think I'd rather he give it because he did the computing.
Right.
Okay, but we did some calculations showing that it's reasonable for 2 billion years to have occurred before a fusion was possible.
Yeah. Yeah. I don't know if Susan had anything to say about, like, I don't know if she read the paper or if that was the right paper. I know she sent it to me, but.
I sent it to you, and it was mainly the quote about the mitochondria charge density that I was interested in. It's on the after table 1. Mitochondrial genes key to nuclear genome expansion, and it's mitochondria must respond quickly to changes in membrane potential, and the penalty for any failure to do so is serious. The electron and proton transfers of chemoautistic energy coupling generates a transmain potential of 150 to 200 millivolts over the membrane. That's huge. That's huge right there. But it's only 5 nanometers across, which is where you get this charge density from.
Okay.
Because then you're at 30 million volts per meter, equal to that of that discharged by a bolt of lightning.
Wow.
That's the quote.
Yeah.
So I'm very interested in that, because maybe you can see what is alive and what is not because of the charge density.
Yeah.
And measure it.
Oh well, mine's pretty low.
Yeah.
Pardon?
Mine is pretty low.
Yeah. Okay.
Anyway, I'm going to look up this quote. It's reference 49. So
Yeah.
it's Biddle and Falkowski.
Okay, I'm going to throw out an idea for the 2 of you.
Okay.
Consider a paper with the title, Does mitochondrial fusion contradict the symbiotic theory of eukaryogenesis?
Does mitochondrial fusion?
And we can go back to, what's his name, Dobzhansky or somebody. Everything in biology only makes sense in the light of evolution.
Yeah.
It contradicts what?
Eukaryogenesis by symbiosis.
Yeah.
Think about that.
Yeah.
Okay, that could make quite a splash.
Yeah.
Because that theory is so hard now.
Right.
It might be time to tear it apart.
Yeah.
Okay.
Yeah.
And if it got published with my name on it, Dr. Sharif would have a hairy fit.
That's okay. You get a Nobel Prize.
Right.
Needs to go to bed. Anyway.
Okay. Have a good day.
Have a good week.
Bye.
Thank you.
Okay.
Bye.
Send me that image. Okay.
Okay, I will. All right. Thanks.
Okay. Thanks. Bye.
Bye.
Bye.